Thank you. So I'm very conflicted because I'm the founder and CEO of InSilico Medicine. So whatever I say is benefiting the company. You can add me on WeChat if you like. And one of the biggest challenges of healthcare is that biology and chemistry are very complex areas, and they are currently disconnected. For AI, one of the major challenges is to get validation in humans rapidly. And right now, you've got a hype cycle in AI. A lot of those advances are pretty credible. So since 2014, AI systems outperformed humans in multiple tasks, including image recognition. So ImageNet championship, deep neural networks won and outperformed humans in 2014. So that's when the hype cycle started. And text, voice, pretty much everywhere. So you've seen the Atlas performing all those tricks. The beauty of those tricks is that you can validate in almost real time with your own sensory systems. So whatever AI produced, you can validate in imaging and text. When it comes to drug discovery, the only credible claim you can make is after you pass phase 2 clinical trials, after you show efficacy. And by that time, you need to be to ensure that the drug is safe and also that it's effective, that you manage to implicate the target in a disease properly. So one of the best slides, everybody is using it in pharma, is by Steve Paul, how to improve R&D productivity in the pharmaceutical industry. So you can see the multiple cycles and multiple steps you need to take in order to get the drug to the market. And very often the people who are making the bullets at the very initial stages are not the people, I mean almost always, are not the people who are taking those bullets to the market and aiming them at specific individual patients. The failure rates at every step are dramatic. So you actually fail much more often than you succeed. And that represents a huge problem for artificial intelligence as well. So, and that's actually after you identify the target. So before you start target to hit process, you need to get the target right. And you need to implicate it in the disease. So this process takes a very long time. It's also very disconnected. And today I actually was encouraged not to talk about chemistry. But I think you cannot not talk about chemistry when you're talking about clinical development. So I will. Back in the days, in the early 90s and the 80s, people were talking about which martial art is important and what type of martial art you should study in order to win in a street fight. Should you do boxing? Should you do grappling? Should you go into judo, jiu-jitsu, or something else? So, and they came up with a concept of MMA, mixed martial arts. So where you actually have to be good at everything in order to succeed. And there are some world champions that are amazing at grappling, wrestling, kicking. So I think that in drug discovery, you also need to be good at everything in one go. And artificial intelligence can help you do that. So you need to start with a really good target ID system that can be rapidly validated with CRISPR knockouts or other techniques, and also real chemistry. If you don't get your chemistry right, you're going to fail in phase 1 always. If you didn't get your target right, you will fail in phase 2. So it's important to work with omics, text, biomarkers very early before you go into clinic. And actually what Mike has done at Biotime and at Ajax, so that was the first, to my knowledge, application of deep neural networks to target identification, period. So back then, we've created essentially the cellular differentiation clock based on a huge data set that he's assembled in a controlled environment, and then deconvoluted that clock into targets, and one of those targets was COX7A1. Whether it's going to be a clinically relevant target, time will tell, but it's a very cool exercise. So you need to do a lot of those exercises to do target ID and then validate. I think these processes have to be connected. So if you don't have them connected, you will be failing in phase 2 all the time. And at InSilico, we started working on kind of an end-to-end drug discovery pipeline, which is essentially driverless drug discovery. And before I started working in the pharmaceutical industry, I was much more optimistic about how things are going to pan out. Now I see that we fail all the time as well, and you need to fail a lot to get your to even understand what works in what disease modality. So we have multiple deep learned systems for target ID. So some of them start with aging clocks, and then you deconvolute those into targets. You do causality inference, identify most important features, and you make a target hypothesis. Then you go into virtual screening using AI, or what we're specialized in at InSilico is generative adversarial networks and reinforcement learning. So we combine those two techniques. It's essentially AI imagination and AI strategy. First proposed in 2014, and we use that for both target identification and also small molecule generation. So now we can generate small molecule chemistry with specific properties. That allows you to validate your target hypothesis faster. We also do clinical trials outcomes prediction based on multiple modules. So one is we implicate the target in the disease and see if the products in clinical trials are correctly hitting the target in a specific disease modality, then we assign a specific probability to the clinical trial, and then compare it to the real outcome. So typically it's predictive systems where we wait until the clinical trials succeed and see if our guesses were right. So currently it's piloted by several pharma companies and also investment banks. And one case study I want to talk to you about today, so we just published it last month. It was a pretty famous paper. So here we used generative tensorial reinforcement learning systems to generate molecules with the desired chemical properties. So essentially, you probably have heard about deepfakes, where you can generate faces that are not real. So we use this technique a lot for biological data generation, but here we use it for chemistry. So we were challenged by our pharma partner to come up with novel inhibitors of DDR1 kinase, a kinase implicated in fibrosis and also in other diseases, as we predicted to be implicated. So it took us just 21 days using AI to compile data available around this target after it got presented to us, very quickly generate just 6 compounds that were very easy to synthesize. So it took us 10 days to synthesize those, and 4 were hits. So typically for this kind of exercise, you start with hundreds, if not thousands, of molecules. Here we did just 6, and 4 were single-digit nanomolar hits. So due to budget constraints, we just progressed 2 of them into metabolic stability assays, microsomal stability assays, and submitted to Nature Biotech. One of the reviewers had doubts, so we actually progressed one of those molecules into animals and confirmed really good PK, and then confirmed efficacy as well. So that shows you, and that gave a lot of confidence to computational chemists using this approach. So we can now do things like that very quickly, and it allows you to validate your targets very quickly, so increasing the probability possibly for successful clinical trials outcomes. So my dream for the next couple of years is to do something like this, so do a CureGo for a rare disease that kills quickly, where we would take 2 months to validate, to get the target validated also, generate chemistry, get into IND, and then go into phase 1, phase 2. Again, for something quick where we would be able to use even compassionate care approaches. So that's our current dream. So we're looking for a pharma partner to take this forward later on. And I think that AI is the glue which can help you link drug discovery and clinical development.
Great. Thank you, Alex.